10 or 9:10, depending on where you are in the world. And so welcome everyone to MVZ Lunch. This is our last one of the semester. I have just a few quick announcements before I turn it over to Chris to introduce our speaker today. The first is, I think everybody's probably seen this, but campus, so the state of California is expected to open up June 15th. Campus is planning to allow everyone who wants to work on campus to be back as of June 16th. And by mid-July is expecting all faculty and staff to be back and working in person. So if you, for any reason, need accommodations, you need to speak with your supervisor about that. But the goal is to get everybody back on campus by mid-July. The MVZ has been approved for phase 4 recovery, meaning that more people are now allowed on campus. So for the students, I sent out an email about this earlier in the week, but you can now come in and work in your offices, following all the usual guidelines of being masked and 6 feet apart, with no more than 3 students per shared office. So we're not going to have sign-up sheets for that, but you can come in and knock on the door, and if there are 3 people in there already, you have to find another place to work. The GML is available, but otherwise you can work in your offices. We have more space in the labs, both the prep lab, the HERP lab, and the EGL. So we're using sign-up sheets. We're continuing to use sign-up sheets for those spaces, but we can accommodate more people. So everything is progressing very nicely. We're still in the orange tier, but apparently I heard this morning in a meeting that things are looking like we'll move to the yellow tier. So yellow is better than orange. So everything's going in the right direction. Stay safe. I will turn it over to Chris now to introduce our speaker today.
Great, thank you, Michael. So it's such an honor and pleasure today to introduce Dr. Katie Peichel, who is currently professor and head of the Division of Evolution and Ecology at the University of Bern in Switzerland. She also was recently elected to the American Academy of Sciences and has received numerous other honors, including Guggenheim Fellowships. But Katie actually grew up in LA and started her career as an undergraduate here at Berkeley, where she majored in molecular and cellular biology right before they founded our Department of Integrative Biology. So you're off the hook. And then she did her PhD at Princeton in the lab of Tom Voigt, where she worked on mouse and the ulnalis gene. Is that correct? And then started her postdoc in David Kingsley lab, where she first worked on Stickleback, and then started her own lab at the Fred Hutchinson Cancer Research Center for several years before moving in 2016 to University of Bern. And if you've ever heard of Stickleback as a model for genomics and speciation research, I mean, Katie has been the driving force on so many of the foundational papers in this system. I mean, and she still is. I mean, it's just really impressive how many projects she's been a part of. So she was the first one to estimate a QTL map, including identifying a QTL for EDA for the armor plate loss in Stickleback, as well as in so many other papers, including the neo-sex chromosome, including sort of right now refining the chromosome assemblies, as well as sex chromosome assemblies. But throughout her whole career, she's also had a fascination with the genetic basis of behavior and has developed many innovative approaches for looking at and phenotyping behaviors that no one else has really been thinking about how to quantify, including schooling behavior, habitat preference. It's been such a pleasure to know Katie over my career. She's done so many sort of innovative, inspiring research that's certainly inspired my own career. And so thank you so much for talking to us today, even though it's 9 p.m. today there. I'm really looking forward to your talk. Thank you so much, Katie. I'll take it away.
Thank you, Chris. That was such a nice and kind introduction. I really appreciate it. I'm really sad I'm not there. I'm just seeing all these faces of people who I would love see and chat with and hug and have a beer with. So I'm really happy to have the opportunity to speak to you today. So let me share my screen and go into presenter mode. So yeah, so today I want to tell you about sort of the work we've been doing over the past decade or so to think about this question of how predictable is evolution. And so this, as many of you know, in this wonderful book by Stephen Jay Gould, Wonderful Life, which actually my mother gave me for Christmas when I was an undergraduate at Berkeley. And I remember I spent the whole Christmas break reading this book. And I think it somehow maybe inspired me a little bit to become an evolutionary biologist, even though I took a detour into developmental genetics for my PhD. And in this book, of course, Gould raises this question as if we sort of went back in time and replayed the tape of life, would evolution repeat itself? Would we have evolved? And the answer to this by Gould was a resounding no, right? The contingencies played a huge role in the evolution of life as we know it. And so that if we did this experiment, life would not be the same. But we know that evolution does sort of repeat itself. And so, for example, the evolution of sort of similar forms of placental mammals and marsupial mammals in response to similar ecological conditions, or the spectacular radiations of cichlids in the Great Lakes of Africa, where you can find the independent evolution of sort of similar forms that have adapted to very similar ecological environments. Okay, so I think, the question, or sort of, it's clear that there are these repeated patterns of evolution. And I just want to lay out sort of some terms here at the beginning. So people use these terms convergent and parallel evolution to talk about repeated evolution. So which is just the independent evolution of similar phenotypes in similar environments. And convergent evolution is in the definition that I'm going to use is where similar forms have evolved from a different common ancestor. So this picture of marine mammals, I think, illustrates this nicely, where you have the independent evolution of manatees, walruses, and then dolphins and whales, and they don't share a common ancestor that was marine. By contrast, I'm going to use the word parallel as evolving from the same common ancestor. So for example, dolphins and whales had a common ancestor that was marine. And so we sort of call this parallel evolution. Beyond the terminology, what's important is that the repeated evolution of similar phenotypes is often interpreted as evidence for natural selection, that you sort of see these same forms evolving in response to similar environments. But I have to say that this assumption has rarely been tested sort of empirically. And so I think still a question is sort of why do we see these patterns of repeated evolution? And this is a question that was sort of explored in another really nice book by Jonathan Losos, where he argues if we can understand why evolution repeats itself or why it doesn't, maybe we can use that information to predict evolutionary responses in the future, which of course has huge implications for medicine, for agriculture, and for conservation, as well as just for inherent interest by evolutionary biologists. Okay, so we're going to explore this question of why evolution is repeatable in the beautiful Pacific Northwest. So I'm showing you here a drone picture of a lake on Vancouver Island and its connected stream. And this is to sort of highlight the difference in these two environments. And within these lakes and streams across the Northern Hemisphere, you can find these so-called lake stream pairs of sticklebacks. So you can find in these small, slow-moving streams that are heavily vegetated, stream sticklebacks, which tend to be sort of deeper bodied and are adapted to feeding on macroinvertebrates in these stream habitats. And then in the adjoining lakes, you find fish that are much more streamlined. So they have a more sort of a less deep body, longer body, and they're generally adapted to eating zooplankton in these lakes. So we find these lake stream pairs actually across the Northern Hemisphere, not just in the Pacific Northwest of North America, but we also have lake stream pairs here in Switzerland. And, you know, for the most part, you see that the stream sticklebacks are sort of deep bodied. The lake sticklebacks tend to be more streamlined. But we don't see that they're perfectly parallel. So stream fish tend to resemble stream fish more than they resemble lake fish. But of course, there's lots of differences among stream fish or among lake fish. So we wanted to sort of understand what was driving these patterns of parallelism. And so the work I'm going to tell you about to start was a collaboration between Dan Bolnick, who's now at the University of Connecticut, Andrew Hendry at McGill, Rowan Barrett at McGill, and myself. And the question we asked is whether we could predict these patterns of parallelism that we see in nature. And so to do so, we assembled a huge field crew, and this work was led by an amazing postdoc in Dan's lab, Yoel Stewart. So now many years ago, we decided to focus on Vancouver Island. And so we chose 16 of these lake stream pairs on Vancouver Island. And we collected as much environmental data as we could across these different lake stream pairs. So we had about 94 different variables across these 32 sites, collected in as consistent a way as we could. We had data, of course, from the fish themselves. So 86 different traits, about 40 fish per each of these 32 sites. And then we also had genomic data, so double digest rad-seq data. Okay. And so the first thing I want to show you is that these 16 pairs are actually independent. So the letters here on the map indicate the locations. These are all from independent watersheds. So we knew that they should be sort of independent, and using the rad-seq data to make a phylogeny, including a bunch of other populations from Vancouver Island and these marine outgroups, what we see is for the most part, a lake and a parapatric lake and a stream are each other's closest relatives. So for example, Misty Lake and stream are more closely related to each other than they are to other systems. So I think for 13 out of the 16 pairs, that was the case. But clearly each of these lake stream pairs was an independent pair, giving us these replicates for our experiments, for our analyses. And then we asked, okay, so, you know, we see these traits that we see stream fish look more like stream fish, lake fish look more like lake fish, but across these traits, how parallel were they? And we did models where we looked at the effect of habitat, so lake versus stream. So a large habitat effect would be highly parallel across these 16 lake stream pairs. And then a habitat by pair effect, a large effect here would mean that there was some effect of habitat, but there was also sort of differences among the independent pairs. And so I have these different traits are colored here for just different types of traits, trophic traits, swimming traits and defense traits, and then traits we didn't, mostly shape traits. So now I'm going to show you sort of individual traits and sort of what those patterns look like. So these letters here highlight those. So traits up here would be very parallel. Traits over here would sort of have an interaction between the pair and the habitat, and then traits down here would be sort of not very divergent. So for example, gill rakers, which are the structures that fish use to process food, and we know that fish that are eating benthic resources versus limnetic resources have differences in the number of gill rakers. So in fact, lake fish tend to have more gill rakers than stream fish. And so each of these lines connects a lake stream pair, and the black lines here indicate where the direction is basically of divergence is the same. So for 12 or 14 out of 16 pairs, the lake fish have more gill rakers than the stream fish, and the green indicate cases where that wasn't true. Body depth is another trait that's highly parallel, although there are some differences among populations. So as I suggested at the beginning, streamfish in general have much deeper bodies than their associated lake fish, but there are a few populations where this changes. So these traits are highly parallel. And interestingly, these are the two traits that sort of people already knew about that seem to distinguish these lake and stream pairs. But it was sort of a question for us of whether there were other traits that show these highly parallel patterns. There's also a lot of traits look like this, where sometimes the streamfish have sort of larger values, sometimes the lake fish. So this would be a case where there's a strong habitat by pair interaction, and then traits like plate counts that just don't really vary very much between lake and streamfish. Okay, so we can see these differences in how parallel traits are. We also sort of threw all of the data together and sort of asked about overall levels of parallelism between the systems. So these are just taking all of those 84 traits, doing a principal component analyses, and then plotting sort of the values in principal component space for a lake and its cognizant stream here in green. And then we have these lines sort of connecting them. So, okay, so there's sort of a mess here, right? So we can see that we don't see that all of the lake fish are over here and all in principal component space, and all the stream fish are over here. It's sort of a big mess. You can see maybe these two pairs show very similar. The lakes are very close to each other. The streams are sort of over here. Can we make sense of these multivariate data? And so really Dan and Yoel use this explicitly geometric model to look at these data. So let's say we have lake stream pair A here and lake stream pair B here. You can sort of see that these are very parallel. So the lake fish have very similar phenotypes and the stream fish are very similar, and the lines connecting them are in fact quite parallel. So for any given pair, any given two sort of sets of populations, you can measure this angle between the lines theta. So that's sort of the degree of parallelism. You can also measure the degree of, or the length of these lines. So that's sort of how differentiated the lake and stream populations are from each other. And then for any two pairs of populations, you can make this measurement delta L. So for example, pair D is not very differentiated. Pair A is highly differentiated. So sort of the difference in the amount of differentiation between lake and stream is this delta L measure. And so I'm just going to give you sort of the upshot of these analyses and the final conclusions, which is that we can actually predict the patterns of phenotypic parallelism that we see. So that angle or theta of morphological divergence is actually strongly correlated with ecological divergence. So the degree of morphological parallelism is driven by natural selection. And so what that meant is that basically if lake fish, if the divergence between lake and stream fish was parallel, it's because sort of the ecological divergence between lake and stream was also parallel. So maybe that's an intuitive result, right? That we think that these patterns of parallelism are driven by natural selection, but I think this is one of the first studies to sort of show that, demonstrate that with empirical data. And so then we did not see that sort of genetic divergence or genomic divergence was correlated with this degree of morphological parallelism. However, the actual magnitude, so that delta L, was correlated with the amount of genetic divergence. And so what this means is that the extent, so how much populations can diverge, is constrained by gene flow. Again, an intuitive result that we sort of know. But this was sort of nice to see. So populations that are highly divergent from each other experience very little gene flow, and populations that are not as divergent from each other have much more gene flow. Okay, so this is sort of, I think this was this was a fun study and sort of gave us these satisfying results about sort of these patterns of phenotypic parallelism. But now I want to turn to a question that's sort of closer to my heart. And the question that I've really been thinking about for a long time is sort of, oh, sorry, before I get there, I wanted to come back to these two books. So why is evolution repeatable? So repeated phenotypic evolution does indeed reflect the action of natural selection. But I would say contingency, and in this case, genetic constraints in the form of gene flow, also play a role in the patterns we see. So there's these interactions between selection and these contingencies or constraints that lead to the patterns we see in nature. Okay, so now to the question of the genetic basis of repeated evolution. And sort of we know from classic work done by Michael, of course, on these pocket mice, where we see that the same genes seem to be involved in the evolution of similar phenotypes. So this is a sort of iconic example where the evolution of pigmentation differences across a large number of both mammals, birds, and even fish are driven by changes in this melanocortin-1 gene, right? So this is just one example where we see this repeated evolution at the genetic level. But I've been interested in addressing this question as well. And so I think the question is, we have all these examples of repeated evolution at the genetic level, but how often is this true? So how often do the same genetic changes underlie repeated phenotypic evolution? Can we look at this in an unbiased way? And then if so, why might the same genetic changes be used more frequently when similar phenotypes evolve? And I'm going to try and address both of these questions today. Okay, so again, we're going to talk about these freshwater stickleback species pairs. So these lake stream pairs that I already talked about, the data from these 16 pairs on Vancouver Island, and contrast that with repeated evolution of these so-called benthic-limnetic species pairs, which have evolved in the same region of the world, but in a more restricted set of locations. And I'll briefly introduce these different populations. So I've already introduced you to these lake stream pairs where we have fish in the streams adapted to feeding on macroinvertebrates, and fish in the lakes largely adapted to feeding on zooplankton. And again, this work was done in collaboration with Yoel Stewart, Dan Bolnick, and Andrew Hendry. And then in collaboration with Dolph Schluter, we've addressed this question in these benthic-limnetic pairs, where within a few lakes, also on Vancouver Island and nearby, within single lakes, we have two species of sticklebacks that inhabit these lakes, these so-called benthic sticklebacks that are living in the near shore and also feeding on macroinvertebrates, and then limnetics, which are living in the open water and feeding on zooplankton. Okay. So we've taken a couple different approaches to addressing this question about the repeatability of evolution at the genetic level in these systems to address the question of how often the same genetic changes underlie these repeated evolution of repeated phenotypes. So the first approach is using quantitative trait locus or QTL mapping to find the genotypes that underlie parallel phenotypes, or really regions of the genome. So in this approach, we start with fish that are divergent, lake versus stream, benthic-limnetic. We cross them in the lab, and then we generate large mapping populations that allow us to look at the associations between genotype and phenotype to identify regions of the genome that show these associations. Ultimately, we'd like to identify, sorry, my dog is barking, Debbie, the genes that underlie these associations and the molecular changes. But today I'm really going to be talking about identifying regions of the genome or these QTL that are associated. Okay. So we've done this now for 4 of the 16 lake stream pairs that we studied in the larger study. So they're highlighted here on the map. This work was done by Hilary Poor, who was a technician in my lab and actually helped me move my lab from Switzerland to Bern, as did Sophie Archambault, who is in the audience today. So these 4 pairs are shown here on the phylogeny. 3 of them fit this case where they're each other's closest relative. Pi is sort of an interesting system. We're not really sure what's going on here. But we chose these 4 pairs because 2 of them showed highly parallel patterns of phenotypic divergence with each other, and then 2 sort of show these sort of different patterns of phenotypic divergence. So we were interested in contrasting those systems. And I'm not going to belabor the details here, just give you the punchline, and is that we found that, in fact, few or really no lake stream QTL were shared across these 4 pairs. So I'm just giving you one trait here, which is total plate count. This is showing the chromosome, the 21 Stickleback chromosomes, and then on the y-axis are these LOD scores, which are just the statistical association between genotype and phenotype. And for each of the 4 systems, we had 2 to 3 crosses, which are indicated in the different colors. And all I want you to see here is that the sort of peaks above the lines are on different chromosomes in each of the systems. And indeed, we don't even find the same QTL in different families from the same systems. So essentially, we find no sharing of QTL, even though we see in some cases highly parallel phenotypes across these systems. So this was really surprising because when we did the same thing for a benthic-limnetic system, here we just used 2 different benthic-limnetic pairs. We found that almost half of the QTL were shared. So in this study, we found that 48% of the QTL we identified were found in both of the crosses or both of the lakes. Nearly half, or just over 40%, were found in one lake but not the other. And then there was a percentage of QTL where we found a QTL at the same location in the 2 different lakes, but they had effects in the opposite direction. So the benthic allele made them large in one cross and small in another cross. But still, quite a dramatic difference in the sort of sharing of QTL between the benthic-limnetic system and the lake stream systems. We've also looked at this by doing population genomics. So this is work that was led by Diana Renison when she was a postdoc in my group. She now has her own lab at UC San Diego. And so Diana asked the question of sort of what genotypes are associated with adaptation to these divergent environments. So for the lake stream data, again, we use these same 16 lake stream pairs. We had this DV-RADseq data. For the benthic-limnetic data, Diana had data from 3 benthic-limnetic pairs, and this was GBS data, so not totally comparable, but I'll show you how we dealt with that. So then what Diana did is she made 50 kilobase pair windows and calculated FST between each lake stream pair or each benthic-limnetic pair. And so we had about 2,500 windows in the lake stream pairs and 5,700 windows in the 3 benthic-limnetic pairs. And then she just simply identified the top 5% outlier windows from each of those. And parallel windows in this case were identified as those that were outliers in 2 or more pairs, and non-parallel windows would be either an outlier in only 1 pair or not an outlier at all. And this is important later. Okay. So what do the data look like? So for the benthic-limnetic systems, we basically see these highly repeatable patterns. So again, here's the 21 Stickleback chromosomes. FST is shown on the y-axis. And just visually, what I want you to appreciate here is that the patterns look very similar. So where we see high FST in one population, one pair, you see it in the others. And again, this contrasts really strongly when you look at the 16 lake stream pairs, where we see very different overall levels of FST is low in some populations, high in others. But if you just sort of visually look here, and we quantified this, I'll show you, but you don't see the sort of striking pattern that you see in the benthic-limnetic pairs. And so, for example, if you just plot any two of the population pairs, FST, so for the benthic-limnetic pairs, you know, if a window is an outlier of high FST in one population pair, it's a high FST in the other. So the correlation for benthic-limnetic pairs was 0.6. Whereas the correlation for the stream lake pairs was somewhere around on average of 0.15. And there's also almost no correlation between the stream lake data and the benthic-limnetic data. So a question that I would love to discuss with people is sort of why there's such this dramatic difference between the systems. Is it the source of standing genetic variation? We think that freshwater stickleback populations come from standing variation in the marine population, but these are all from Vancouver Island. The source of standing genetic variation, the marine population should be pretty much the same in all of these systems. Is there something about the evolutionary history of these systems that is driving this? Again, that's something that we can discuss. Something about the extent of gene flow. So there is very strong gene flow between benthics and limnetics in these systems, but gene flow is highly variable in the lake stream systems. That's something I'd like to explore more deeply. Do we see higher levels of parallelism among lake stream pairs that are experiencing higher gene flow, for example? There's a really interesting paper by Ken Thompson from Dolph Schluter's lab that suggests that the strength of parallel selection actually influences the probability that the same genetic loci would be used. So the more parallel selection is, the more likely it will be that the same genetic changes are used. So it could be that the parallel selection is much more highly parallel in the benthic-limnetic systems than in the lake stream systems. So again, I don't have answers to these questions, but just there are things that I would love to get people's ideas on, and something I've been thinking about a lot. Okay, so in the last bit of the talk, I've sort of shown you the patterns. So I've said sort of sometimes we see the same genetic changes, and again, same genetic changes being defined as QTL or genomic regions, underlie repeated evolution, but not always. And this is also true in Stickleback now in sort of case studies when people have gone in, people like Craig Miller, who's in the audience, have looked at specific phenotypes. Sometimes we find the same genes, but not always. So now the question is, for me, is why? Why is it that when the same genetic changes are used, when the similar phenotypes evolve, why is that? And there's some possible explanations. So one is that there's really only one gene or one mutation that can produce a phenotype that's favored by selection. This is probably not the case. So we know, for example, in the case of pigmentation in mammals, that there's hundreds of genes that, if mutated in the lab, can lead to changes in pigmentation. But there's only relatively few genes that seem to be involved in evolutionary changes in pigmentation in nature. So this option is probably not the case for most phenotypes. The second option is that there are many genes or mutations that can produce the phenotype, but that some may be less pleiotropic. So if a mutation affects very few traits, therefore there might be fewer fitness constraints. So there's less likelihood that that mutation is going to have negative pleiotropic effects. It would have its sort of beneficial effect on one phenotype, but then less possibility that it would have negative pleiotropic effects on other phenotypes. The second is that maybe some genes just have higher mutation rates, and there's, interesting study, case study of this in Stickleback. Again, beautiful work from David Kingsley's lab showing that repeated mutation at the PIDX1 gene leads to repeated loss of these pelvic spines, and that this locus actually has very high mutation rates. It's also possible that regions of the genome that have low recombination may be more likely to contribute to these adaptive phenotypes. So we set out to try and test some of these ideas. And again, this was work that was led by Diana Renison. And so she used this genomic data that I already described to you, and we sort of focused on whether we could identify differences between these parallel windows, which were outliers in 2 or more of the pairs, versus these non-parallel windows, which were either outliers in only 1 pair or not outliers at all. And I'll just say that we did the analyses both ways. So we just we just took all the outliers and we said, was it non-parallel or parallel? And then we also did it sort of including non-outliers, and the results are qualitatively the same. So just to give you again a sense of the data, so this is for the benthic-limnetic data. We had 172 of these windows that were differentiated in 2 or more pairs, and then there was many windows that were not differentiated at all, and then 405 of these non-parallel windows, which were outliers in only 1 pair. So we're really comparing sort of these windows to these windows that were outliers in 2 or 3. And then again, for the stream lake, even though I showed you it was much less parallel than the benthic-limnetic system, we still find many windows of the genome which actually are quite parallel. So like these guys are outliers in 9 or 10 of the populations. But again, we had a loose definition here for parallel, so it just had to be differentiated in 2 or more pairs. And then we compared this to the non-parallel windows that were only differentiated in 1 pair. And then we asked, do they differ? Do they differ? So in the level of pleiotropy, so of course, to estimate pleiotropy, the ideal test would be to make a mutation, to make all possible mutations in a gene, and to measure all possible phenotypes as well as the fitness consequences of that. But of course, that's sort of a challenging experiment to do unless you work maybe on yeast, maybe C. elegans. Oh, and of course, then test that in multiple different environments. So we used a couple of proxies. So one is David Marquez and I had compiled a big database of all the QTL studies that had been done in Stickleback. So we have about 1,000 QTL that have been identified now for many different traits in Stickleback. So we use that as sort of one level of pleiotropy. If there's multiple QTL, it suggests that maybe that region of the genome is pleiotropic. We also used RNA-seq data, so looking at whether genes were co-expressed or not. And so it's been shown in other systems that genes that are sort of highly co-expressed with many other genes are highly connected, tend to be more pleiotropic. So these were sort of our 2 independent measures of pleiotropy, each of which comes with its own set of caveats. But the idea was to sort of use these 2 different independent measures to see what the results would look like. Then we also asked whether these windows varied in gene number or in recombination rate. And I'm just going to show you a few results and then give you sort of the summary, and also in mutation rate. So here was the first striking result. So contrary to our hypothesis, so we found that parallel windows contained more QTL. So for the lake stream pairs and the benthic-limnetic pairs. So this is showing the number of traits with mapped QTL in a window. And so it's hard to see here, but the parallel windows, I should put the numbers here, have about twice as many QTL as the non-parallel windows. And that was true in both the benthic-limnetic and the lake stream pairs. So this is sort of, again, contrary to that hypothesis, we would have predicted that parallel windows should have fewer QTL, they should be less pleiotropic. But interestingly, in the benthic-limnetic data, you can sort of see that the non-parallel windows actually have the most QTL. So there are some of these non-parallel windows that seem to have many, many QTL mapping there. We saw a very similar result for the gene connectivity data. So again, the parallel windows tend to have genes that are more connected in both the stream-like and the benthic-limnetic. It didn't quite reach statistical significance for the benthic-limnetic, but that the non-parallel windows actually had more, the most highly connected genes. Okay, so the level of pleiotropy seemed to be that the parallel windows had more QTL and higher connectivity, suggesting that they were more pleiotropic. But the non-parallel windows seem to have the most highly pleiotropic regions. Gene number did not differ between the parallel and non-parallel windows, and recombination rate did differ in the benthic-limnetic system, which, as I mentioned, has very high gene flow. And you can sort of see that pattern I showed you, that you see very high FST in regions of low recombination and sort of low FST in regions of low recombination. So, but when we looked at this, when we looked at this in the only in outlier windows. So all of the outliers are basically in these regions of low recombination. But then if you compare parallel versus non-parallel windows, you don't see a difference in recombination rate. Hopefully I said that in a clear way. So basically, in outlier windows, this difference in recombination rate sort of goes away. So we don't think that that's a key component here. And then mutation rate also did not differ, especially if you account for the difference in recombination rate. So really the main result here is the difference in levels of pleiotropy. Okay. So I've shown you that sometimes we see the same genetic changes or same genomic regions underlying phenotypic evolution, but not always. But when we do see the same genetic changes, we think that recombination rate might play a role when levels of gene flow are high. But we're sort of hypothesizing from these results that it's lower intermediate levels of pleiotropy might actually be beneficial for adaptation. So contrary to this idea that pleiotropy is always a constraint, it may only be constraining at high levels, and sort of lower intermediate levels might actually facilitate adaptation because, you know, sticklebacks adapting to lake versus stream habitats have to evolve many different phenotypes that make them a good lake or a stream stickleback. And particularly in systems like Stickleback, where a lot of the variation is maintained as standing variation, old standing variation, these loci might have been tested. So these loci that have this sort of perfect amount of synergistic pleiotropy might be maintained over long evolutionary time. And then as sticklebacks sort of repeatedly adapt to similar environments, these sort of perfectly pleiotropic loci might be the ones that are repeatedly used. So that's sort of what we're thinking about now. But of course, again, I'm super happy to discuss. So in the last, I think I have 5 minutes is that okay, Chris? Okay. So this question of sort of why do lake and stream paras differ, you know, the problem here is we're looking at modern populations and we don't know what the founding populations were, and we don't know how the ecosystem might have changed over time. So our sort of knowledge of what we're sort of taking this backwards looking approach as we mostly do in evolutionary biology. And so if there were sort of contingencies at the beginning that might explain these patterns, we wouldn't be able to see them. So how would we actually go about this? You know, what if we could do Gould's thought experiment and sort of go back and repeat the evolution of these stickleback pairs and try and get at this question of whether there's contingencies that are present that are sort of biasing the direction of evolution. So yeah, I mean, basically we'd want to conduct these forward in time experimental evolution studies in which we know the properties of the founding populations. And then we can ask whether these replicate populations follow deterministic trajectories or due to natural selection, or are there contingencies that predominate these trajectories? And of course, these experiments have been done, right? So there's these, of course, the beautiful experimental evolution work by Lenski and many other people that have sort of addressed these questions beautifully in these experimental systems. But most of these studies have been done in the lab. So the question that we asked is, what if we could do this experiment in nature? Wouldn't that be cool? And we actually have had the opportunity to sort of start this experiment. So I just want to tell people about it. I'm super excited about it. We have nicknamed it Fitness, the Forward in Time Eco-evolutionary Study of Selection. So we had this chance because there were 10 lakes in Alaska that were poisoned with rotenone by Fish and Wildlife to eliminate invasive pikes. So everything was killed in the lakes. And then we had the opportunity to actually put sticklebacks in these lakes. So this was organized by Andrew Hendry. And we were able to collect baseline data from both the source lakes that we used and the recipient lakes. And so, yeah, the plan and the introductions worked. So all of the lakes actually contain stickleback populations in spring of 2020. So despite COVID, luckily a couple of people were in Alaska and able to collect both phenotypic and genetic samples in 2020. And there's a small crew going this spring as well to collect. So the plan is to follow these populations basically from genes to ecosystems for the next, decades, which is pretty exciting. So I won't go into the details here for the interest of time, but the idea is basically to track changes in phenotypes, in genotypes, and in the ecosystems themselves, to work with theoreticians to sort of see how these populations are evolving over time, and whether we can sort of understand the relative roles of deterministic processes and contingencies in these evolutionary trajectories. So I'm super excited. It's going to be a lot of fun once we can actually get back into the field, especially, and we can get the money to do all of the sequencing of 10,000 sticklebacks, which went into these lakes. So with that, yeah, I'm super happy to take questions. I just want to thank my collaborators, which are many over the past many years. I think I've mentioned everyone, but especially the folks I worked with on the benthic-limnetic pairs, together with Dolph, and Dan and Andrew, Rowan and Yoel on the lake stream pairs. And then this fitness project is being led by Andrew, and he sort of roped in a large collection of us to take on. This is a collection of aquatic ecologists, people like me, genetics and genomics. Matt Walsh is interested in the invert communities. So it's been a really fun collaboration that we are looking forward to. So I will stop there, and hopefully there's some time for questions.
Great. Let's all unmute and give Katie a round of applause.
Thanks.
Questions? Okay, I'll jump in. Yeah, fantastic talk, Katie. I think, I mean, and that contrast is still so fascinating too. I guess one of the questions I had was sort of detectability, and just thinking about, I mean, you finding maybe the detectability of the parallel windows. I guess I'm wondering too, so what we're seeing in our pupfish data is if we look for the same QTL across our crosses with the same sort of significance thresholds, we'll find one QTL in one population, but not the other. But then if we relax our thresholds for the other population and just look for any kind of log peak there, we do find a peak. I'm just wondering how much the sort of detectability of QTL as well as QTL effect size, and how do you think that plays a role in all these patterns?
I mean, yeah, this is always the question is like, is there really not a QTL there? Or did you just not detect it? At least for the benthic-limnetic study, we had pretty similar, we had the same sets of marker. These were SNP arrays. So we had sort of similar sets of markers, similar cross sizes. And we did, there was a nice framework in that study. So for trying to address just that question. In the Lake Stream study, you know, we had the perfect plan of it was going to have the same number of individuals, blah, blah, blah. And of course, some of the crosses are bigger or smaller. But I was just looking at this today because I'm trying to work on the paper, but you don't really see sort of the number of QTL is not scaling with the number of individuals in the cross or the number of markers. And we haven't really tried to relax the law threshold or anything like that. But I mean, this is always the challenge. But I think to me, it's really striking. Like there's really, and even different crosses from the same set of grandparents, or from the same watershed. So, and that's the dirty secret. The dirty secret of QTL mapping is that we almost always use one set of, you know, you take like this benthic dad, this limnetic mom, you make your cross, you do your thing, you write your paper, but hardly anyone does repeated crosses from the same populations. And I think there's a lot of segregating variation as well. So I don't know, Craig might have something to say about that too, but yeah. But the contrast too between the systems is really striking. I don't think it's just sort of the sort of a power of QTL mapping or whatever that can explain that.
Awesome talk, Katie. That was great. I had a question. I remember Dan Bolnick in the last few years talking about spine fragments in core samples to get at the question of whether the ancestral populations might have been similar or different. Is there any hope for ancient DNA?
Well, actually, there's a paper in Current Biology that just came out on ancient DNA and sticklebacks. Or maybe it's still a pre-print, but Felicity Jones and Andy Foote have a paper where they got cores from Norway. I think they're from Norway. And they can see some of this ancestral variation. And it looks like a lot of stuff is just lost by drift.
So for the benthic-limnetic lakes and the lake stream lakes, would it be possible to get those lakes scored?
Yeah, that would be super cool. Oh yeah, thanks Noah. Put in the chat, put the paper. Yeah, that would be cool. That'd be cool. I'm not sure if we can get cores from those lakes, but I think that'd be super cool.
Katie, that was great. I had a question that was, you know, a little bit similar to what Chris was driving at. So to get away from the issue of focusing on just the significant QTL, what if you were to construct polygenic scores for each cross and then ask how predictive those polygenic scores are for phenotypes in other populations? It just seemed like another way of getting at, you know, the genetic basis for traits that are almost highly polygenic and where you're not going to detect genes unless they have large effects.
Well, that's an interesting idea, Michael. Yeah, I mean, I think we can, yeah, that's an interesting idea. So you basically have your collection of sort of, from one population, you sort of estimate like these are the loci that seem to be, or the polygenic score, and does that hold in another population? Yeah,
how predictive is it? I mean, this sort of thing is being done a lot in human GWAS, and you know, your setup is a little bit different, but you know, you might be able to ask about the contribution of genes that don't meet genome-wide significance in
Yeah,
in a
yeah.
PL.
Do you- Is an F2 cross suited for that, or would that be more like a sort of association mapping?
Well, I think more often it's done with association mapping. It probably depends on how, how many informative meioses you have, how many F2s you have, but you could prune your data set, down and still do it, I would think. The other question I had just about the comparison between the benthic and limnetic versus lake stream is, can you give us a feel for the time frame for the divergence of these forms in each case? So are the benthic and limnetic forms, do they share ancestry much more recently than, say, the lake stream pairs?
So that I meant to say that. So basically all of these are post-glacial. So they're all, 12 to 15,000 years old. I mean, of course, now we know these ancestral variants are much older than that, but that should be the same pool of ancestral variation. The one unique thing about what's thought about the benthic-limnetic system, those lakes have been experienced, there was a period of allopatries. So basically marine sticklebacks invaded the lake. Dolph thinks they became benthic, and then there was a couple thousand years later, they're at the elevation that suggests there was a second wave of invasion from marines, and those became the limnetic. So there was some period of allopatry. The evolutionary history of the lake stream is probably these watersheds formed, they got sticklebacks. But it's all post-glacial. It's all the same amount of time. So that also can't really explain the difference. Rosemary has a question.
Yeah, sorry. This is excellent, Katie, very thought-provoking. So just following up on that, I was trying to figure out, I mean, presumably when they go benthic-limnetic, presumably, I know Dolph said that the first one to get there is benthic, right? And but presumably they have a much higher ecological amplitude. And so then, it's more of a character displacement thing that is driving the divergence rather than in the lake stream. It seems like there it's really parapatric. It's a very different kind of mode of divergence that's driving the effect.
That's right, and that's why this paper by Ken Thompson is super interesting, where he's suggesting that it's sort of the strength of parallel selection that that somehow results in more parallel genetic changes. I need to delve more into this paper, but that's sort of the punchline of it. And so I think it's true that sort of in the benthic-limnetic system, you really have very parallel, strong selection, keeping these benthics and limnetics apart. And maybe across the lake stream systems, selection is more idiosyncratic and they're parapatric, so there's not there's some gene flow, but maybe not as much. And so, yeah, I think I'd like to try and delve a little bit more into that idea, and maybe we could look at, which is what we sort of thought. Like I thought two of the pairs we had are super parallel, and but we don't see any difference in the genetic architecture. That was my grand plan, is like we were going to have strong parallelism genetically in these two pairs and not in these other two pairs, but that didn't come out. But yeah. So
Very
I
cool.
think more about sort of how we could sort of tease that apart. But
yeah.
Noah?
I'm muted. I just had thank you for a wonderful talk. It's very thought-provoking. It got me thinking of parallels in Arabidopsis, where there can be long-range linkage disequilibrium set up by selection across the range in response to this biotic environment. And these are in glucosinolate, toxin precursor genes. And I'm wondering, do you ever see evidence of sort of long-range LD that's associated with adaptive phenotypes in these sticklebacks? That's kind of one extreme question. And then the other was kind of the role for, I really like the idea of thinking about pleiotropy, as a constraint, but also the other side of it, I think, is epistasis and the idea that perhaps there's both intramolecular and inter that is, much harder to detect, I think. But I just, those are kind of extreme ends of a continuum. And I'm wondering if you have thoughts on how that, if there's a role for these processes in setting up or driving the phenotypic repeated patterns that you see.
Yeah, I mean, I guess, so for the sort of long range LD, the best sort of data sets to look at this are sort of the marine freshwater comparisons, where we have sort of the ancestral marine with all these ancestral variants that are, or these old variants that are freshwater adapted alleles that are sort of carried at low frequency. And then as soon as the fish go into freshwater, they sort of reassemble in a short amount of time to make a nice freshwater fish. Felicity Jones is sequencing like 10,000 marine sticklebacks and sort of trying to address this question of, which allele do these marine sticklebacks sort of carry? How many of these freshwater alleles are they carrying? Are there fish that have multiple alleles, and those are the ones that sort of go into freshwater, and sort of how that works? If that sort of answers your question.
Well, it was a classic Noah question, which wasn't really a question. It was more just to get you to talk about it, because
yeah, no, I think that's super interesting, is how do these things reassort? Because they reassort really quickly. And
yeah,
I mean, so that's the answer, is we know these alleles are old, so we're not relying on new mutation, but they are assorting themselves really quite quickly. And
yeah, it's almost as if it's predictable at some level, right?
And then, I mean, epistasis is, of course, I'm sure it plays a role, and that could play a role in why we see different QTL and different crosses, but it's just so hard to detect. Because I mean,
even in the simplest things, and like yeast or Drosophila, you can have one mutation that is like gives positive sign epistasis for one phenotype and then has a negative effect on another. It's so hard.
And of course, in all of these cases, we're measuring effects on phenotypes, and that doesn't necessarily directly translate to fitness either. So the fitness effects of these loci, we don't know.
Well, thank you for elaborating.
Yeah, I mean, I'm sure epistasis plays a role.
Yeah.
And
So I have a comment. You addressed the idea of some ice age changes, and one of the major things that's happened has been sea level changes. 125,000 years ago, it was 8 meters plus, and then it fell and stayed below the modern sea level for the next 100,000 years, ending 13,000 years ago at 120 meters below sea level. And this must have changed connectivity of the streams, at least around the edges of the island. And maybe it wasn't even an island at that time. And surely it changed the maybe even vegetation and precipitation during such dramatic changes. And of course, there's also the tectonic uplift of that whole coastal sequence that may have raised the lakes by another, 60 to 80 feet if we compare it to, say, something like Newport Bay in Southern California, which was a tectonically uplifted thing, so that we see the 8 meter thing at 80 feet, 25 to 80 feet in just that 100,000 years. So there are major changes going on. I don't know anything at all about that part of North America.
I mean, I think that's exactly how these sort of freshwater populations were founded, is from the tectonic, the uplift, right? That there was sort of they were underwater, the marine sticklebacks were swimming around, and then you get raising up of the land, and sort of marine sticklebacks are trapped in these newly created freshwater lakes and streams. But what I don't know, and that's what I was referring to in these benthic-limnetic lakes, where you have these two species in the same lake, is that there were 2 times that there was sort of connections to the ocean. But then the sort of how this lake stream systems were formed is something that I'm not super clear on. That's these parapatric lake stream systems. They would have seemed to be all a part of the same event, but that I don't know.
Well, geologists probably along that coast, certainly along California and Oregon, have worked out sea level changes and tectonic changes,
Yep.
so that they can tell you which is which in your situation. You also have submarine records in those areas that might tell you something about the connectivity of the island to the mainland, or whether or not there were any connectivity between streams in an offshore sense.
Yeah, no, we should look into that some more, I think, for those lake stream systems. That'd be interesting.
Well, like Gould said, history matters.
Yeah.
Katie, I remembered the question I wanted to ask you if we still have time for one more, but kind of building on Rosie's question too, but I think you touched on this, but instead of the strength of parallel selection across lakes, I think we know the strength across the lake stream versus the benthic-limnetic. Is selection, like divergent selection, much stronger for benthic-limnetic pairs than lake stream pairs?
Well, that's what I was saying. Yeah, I think it is, but I think it is, but just because I think it is, I think we have to quantify that in some way.
Okay, I thought there was enough experiments out there. Everybody's been doing that. It's out there in the literature, but I wasn't sure.
But the other dirty secret in Stickleback is no one really measures selection in the wild on traits.
It's true.
I mean, Dolph's done a little bit. I mean, there's a little bit, but it's not. I mean, we use these arguments of like, oh, we use the parallel evolution argument to argue for selection, but really going out and measuring the fitness effects of specific traits has not been done very often in Stickleback. So this is something Diana Renison wants to really tackle that question in her own work. So yeah, so I think we can't answer that question quite but I think it's true. So
cool. Really amazing contrast. I just did not realize that. But
yeah, well, and I like to make the point too, because I think now everyone's just like, oh, Sticklebacks, parallel evolution, it's all repeated, selection on standing variation. It's always the same gene. It's EDA. And even though I work on EDA, and my former student Sophie's in the audience, we worked on EDA together. It is not all like EDA, right? There's still, I think, interesting surprises to learn in the system. So
for sure. Well, thank you so much. Another round of applause.
Thank you, everybody. It was super fun.
Thanks, Katie. Great to see you.
Nice to see you all.